Before moving forward, we need to define our terms. The term abiogenesis specifically refers to the concept that life came naturally from non-living chemicals. Some people will try to make the case for abiogenesis by defining it to include the creation of life by God. However, here we will specifically use the term abiogenesis to refer to a totally naturalistic concept for the origin of life. That is, it excludes, by definition, the involvement of any intelligent agency, whether it is God or aliens. Our use of the term is going to refer exclusively to a totally naturalistic origin of life. The best argument in support of abiogenesis is the fact that we are here. However, this requires one of two things. The first is defining abiogenesis to include any origin of life, including divine creation. The second is to exclude divine creation as a possibility summarily and to conclude that naturalistic abiogenesis must have occurred. In either of these cases, they're setting up a situation intended to preclude another possibility. By specifically defining abiogenesis as a naturalistic process, we eliminate this problem along with the argument because of the possibility of intelligent design and divine creation. The statistical problem with abiogenesis is based on the simple fact that calculations indicate that the odds of being able to get just the proteins and DNA for the simplest possible living cell, let alone assembling that cell, exceed the number of possible events that could have occurred in the entire history of the Big Bang universe by tens of thousands of orders of magnitude. When talking in terms of probability, this makes it statistically impossible. In fact, it turns out that to get the probability of getting that first cell on just a single planet to a reasonable level, you have to reduce it to only 8 proteins, averaging only 8 amino acids in length. By the way, even this unreasonably oversimplified cell doesn't include the probability of putting the cell together. The time problem in abiogenesis is one of the statistical problems associated with abiogenesis. It refers to the difficulty of fitting the enormous chemical complexity required for life's emergence into the relatively short window estimated to be available early on Earth. There would have been at most a few 100 million years, and possibly far less, for the spontaneous formation of organobuilding blocks, their concentration, polymerization into RNA or peptides, development of protocells, emergence of self-replication, and the evolution of primitive metabolism. Each of these steps is highly improbable and interdependent, and the required molecular structures, functional RNA strands, catalytic networks, stable membranes, would need to arise multiple times or persist long enough to be selected. Compounding the issue, early Earth would have faced constant sterilizing impacts during what is called the late heavy bombardment, potentially resetting chemical progress. The result is a significant timing challenge. The known geological window for life's appearance seems extremely narrow relative to the complexity of the processes required, making it difficult to model a fully natural origin of life within the available time frame. A common misunderstanding is that abiogenesis has a problem with entropy due to the second law of thermodynamics. However, there is another principle in thermodynamics that is more fundamental than the second law, because it explains how the second law of thermodynamics works. According to this principle, when energy is applied to a system, the entropy of that system is driven towards the entropy of the applied energy. Consequently, if energy is applied to a system that has a higher entropy than the system, the energy tends to increase the system's entropy. If the energy has a lower entropy than the system, it decreases the entropy of the system. A good comparison is that this principle represents the difference between construction work and a bomb. When this principle is applied to abiogenesis, the problem is that there are no prebiotic sources of energy with an entropy level anywhere near as low as that of even the simplest of living cells. As a result, any system that comes close to becoming a living cell without actually achieving will be driven away from it by the available sources of energy. This results in a thermodynamic problem of producing life from non-living substances. This is closely related to what is commonly called the energy problem. The energy problem abiogenesis centers on how the first prebiotic systems could have accessed, stored, and used usable energy in a consistent, organized way without enzymes, membranes, or metabolic pathways. Life today relies on highly sophisticated mechanisms, ADP synthase, proton gradients across membranes, redox reactions, and tightly regulated metabolic networks to capture and convert energy. But before such machinery existed, early molecules would have needed a steady supply of energy to drive polymerization, maintain stability, and support any form of replication. Many natural energy sources, UV radiation, heat, lightning, hydrothermal vents, are either too destructive, too diffuse, or too inconsistent for delicate prebiotic chemistry. Furthermore, polymerization of peptides and nucleic acids requires energy input, yet water-rich environments tend to favor depolymerization, not synthesis. Proposed solutions such as mineral-catalyzed redox cycles, geothermal gradients, or wet, dry cycling remain incomplete and unproven as self-sustaining systems. Until a feasible mechanism is demonstrated that can reliably power the formation and maintenance of early biomolecules, the energy problem remains a central unresolved challenge in origin of life research. Even though chemistry is where most of the actual scientific work on abiogenesis is done, there's still a lot of problems associated with chemistry, including the results of the very research that is being done. Now, it is true that many fundamental building blocks of life have been produced in labs, but there are four major problems with these results. The first is that these basic components of life, while chemically produced outside of a living organism, are being produced under highly controlled laboratory conditions. This is in strict contrast to them being produced under natural conditions. In fact, there's a lot of intelligent design going on to produce what byproducts are created. The second of these problems is the fact that the chemicals that form the building blocks of life make up only a small fraction of the results of the reactions being conducted. The rest of these products are junk that not only hinders further reactions, but also has a tendency to destroy the building blocks that were actually produced. What is necessary to take the process further is to either conduct a lot of filtering, once again applying intelligent design, or by buying a fresh supply of those products that are themselves produced by living things. The third, called the polymerization paradox in abiogenesis, refers to a core contradiction at the heart of origin of life chemistry. The very reactions needed to build biological polymers, like proteins, RNA, and DNA, are thermodynamically unfavorable in the same watery environments where life is thought to have arisen. Polymerization requires removing a water molecule to form each peptide or phosphodiester bond, but aqueous environments energetically favor the opposite reaction, hydrolysis, which breaks polymers apart. Thus, water both enables the chemistry that produces monomers and prevents those monomers from linking into long, stable chains. Even when polymerization is achieved in the lab, it almost always requires highly controlled conditions, activated monomers, or energy-rich chemicals that do not have clear natural prebiotic sources. This creates a paradox. The environment's rich enough in building blocks, warm little ponds, oceans, hydrothermal systems, are also the very environments that chemically inhibit the formation of the long polymers essential for life. Resolving how long information-bearing polymers could have formed and persisted under realistic early Earth conditions remains one of the central unsolved problems in abiogenesis research. The fourth is that nothing that has actually been produced is even remotely close to an actual living cell. The best that has actually been produced are lipid spheres containing some of the chemical building blocks of living things, sometimes including proteins. However, the gap between these and even the simplest of actual living cells is enormous. To make that jump would require setting up a DNA storage and processing system, as well as all of the necessary cellular machinery for a functioning cell, none of which these lipid spheres are even close to doing. This could have been included with chemistry problems, but it deserves its own section. The Homochirality Problem in abiogenesis refers to the fact that all known life uses molecules of a single-handedness, left-handed amino acids and right-handed sugars, while natural, non-biological chemistry reliably produces racemic mixtures containing equal amounts of both forms. This is a major challenge because mixed chirality prevents the formation of stable, functional polymers like proteins and RNA. Even small amounts of wrong-handed monomers disrupt chain formation and folding. No widely accepted prebiotic mechanism has yet been demonstrated to generate a strongly biased chiral environment on a hypothetical early Earth, nor to amplify a small initial imbalance into the complete homochirality seen in life. Proposed explanations, such as circularly polarized light, chiral mineral surfaces, asymmetric autocatalysis, or selective crystallization, remain speculative, often requiring highly specific or rare conditions. Until a plausible natural pathway is identified that produces and maintains significant chiral purity, homochirality remains one of the most persistent and fundamental unresolved problems in origin of life research. Polymerization in water without enzymes is a central challenge in abiogenesis because the formation of biological polymers, proteins, RNA, and DNA, requires chemical bonds that are thermodynamically unfavorable in aqueous environments. For example, peptide bonds and phosphodiester bonds form by removing water, condensation reactions. But water-rich environments naturally favor the reverse reaction, hydrolysis, which breaks polymers apart. Enzymes in modern cells overcome this problem by lowering activation energies and providing precise catalytic environments. But in prebiotic conditions, no such catalysts existed. Laboratory experiments have shown that polymerization can occur under specific conditions, such as on mineral surfaces, via dehydration cycles, or using chemically activated monomers. But these setups often rely on highly controlled parameters that would not reflect realistic early Earth environments. The challenge is how sufficiently long functional polymers could form and persist spontaneously in natural aqueous settings, without enzymes, energy-driven machinery, or controlled laboratory conditions. This remains one of the most significant hurdles in explaining these chemicals. Abiogenesis also has a huge problem with regard to information. The simple fact of the matter is that DNA is known to store gigabytes of coded, complex, specific digital information. However, such information is known to ultimately only result from an intelligent mind. There is no known process whereby the laws of nature by themselves are capable of producing complex, specific information. There are two main ways that evolutionists try to deal with this problem. The first is that they deny that DNA contains any actual information. They will simply claim that it's nothing but a bunch of chemical processes going on and that it doesn't actually qualify as information. This is a ridiculous argument because it does not matter how the information is stored, transmitted, and processed as to whether or not it qualifies as complex specific information. The qualification is, does the sequence encodes for something specific, and in the case of DNA, it does. DNA has been known for decades to encode for the manufacturing of proteins within the cell. In more recent years, it has been discovered that much of this DNA carries out other functions, including regulatory information. The second is that they will try to confuse complex, specific information with statistical information. While these two types of information are related, they are not the same. Statistical information is a way of dealing with the transmission and storage of information without regard to content. Statistical information does not matter whether or not the information has any meaning. With complex, specific information, on the other hand, meaning is the key to defining this type of information. Ultimately, what is necessary to get abiogenesis to work is to get rid of the information source and transmitter. It literally requires taking genetic noise and turning it into complex, specific information. This is because that is exactly what mutations are. It is even worse in a prebiotic system because you do not have any of the information yet. But even if you have DNA or RNA, you have nothing but noise. The claimed solution is natural selection, but this does not work in a prebiotic system because there is nothing to select. Also, without a specific goal, all you are doing is selecting noise from noise. The origin of metabolism problem concerns how the first energy harvesting and molecule transforming networks, precursors to today's complex metabolic pathways, could have emerged without enzymes, genetic regulation, or cellular structures. Modern metabolism relies on intricately choreographed cycles, such as glycolysis, the citric acid cycle, and electron transport chains, all of which require highly specific proteins and cofactors that cannot plausibly arise without prior biological machinery. Yet, abiogenesis requires that some form of protometabolism existed before enzymes and genes. Proposed solutions, like Wachter-Schauser's iron, sulfur world, alkaline hydrothermal vent chemistry, or mineral-catalyzed redox cycles, claim that simple reaction networks could have been sustained by geochemical energy, but none have been demonstrated to operate autonomously, evolve, or transition into biological metabolism. A further complication is that metabolic cycles require many interdependent steps. Removing even one reaction typically collapses the whole system, making stepwise evolution difficult to model. Until a plausible, self-maintaining network of reactions that can operate outside biological cells and gradually evolve toward enzyme-driven metabolism. The origin of metabolism remains an unresolved and central challenge to abiogenesis. The concentration and localization of building blocks is a major problem in abiogenesis because the organic molecules required for life, amino acids, nucleotides, sugars, and lipids, would have been extremely dilute in a vast early ocean. Chemical reactions that form polymers, like proteins or RNA, require relatively high concentrations of monomers and some form of spatial organization. Yet simple diffusion in water tends to disperse molecules rather than bring them together. Without mechanisms to concentrate and localize these building blocks, polymerization and the formation of functional molecules become highly improbable. Various hypotheses have been proposed to solve this problem, including evaporation in small ponds, adsorption onto clay or mineral surfaces, confinement in ice matrices, or entrapment within lipid vesicles and hydrothermal vent pores. While these ideas show potential in laboratory models, no single natural environment has yet been demonstrated to provide the right combination of concentration, protection, and chemical compatibility to reliably drive prebiotic chemistry toward life. The formation of functional cell membranes is a significant challenge in abiogenesis because early life would have required some form of compartmentalization to separate and protect biochemical reactions. Modern cells rely on phospholipid bilayers that are selectively permeable, stable, and capable of maintaining ion gradients, features essential for energy production, replication, and metabolism. Prebiotic experiments have shown that simple fatty acids can spontaneously form vesicles, but these primitive membranes are fragile, sensitive to salts and temperature fluctuations, and do not readily incorporate large biomolecules such as RNA or proteins. Moreover, without active transport or regulatory proteins, controlling the internal environment of a protocell is extremely difficult. The problem is not just forming a barrier, but creating one that is robust, dynamic, and capable of supporting the complex chemistry needed for life. Until a plausible mechanism is demonstrated for the spontaneous formation of stable, functional protocell membranes under realistic prebiotic conditions, this remains a central unresolved obstacle for abiogenesis. The integration of prebiotic components into a single coherent system is one of the most difficult challenges in abiogenesis. Even if individual building blocks, amino acids, nucleotides, lipids, and processes, polymerization, compartmentalization, energy capture, can arise separately, life requires that they work together in a coordinated, self-sustaining way. For example, a protocell would need a membrane to protect its contents, polymers to store information and catalyze reactions, and energy sources to drive chemical processes. Achieving this integration is extremely complex because each component depends on the others. Polymers may degrade without compartmentalization, metabolic reactions may fail without catalysts, and membranes cannot maintain gradients without energy. Experimental models have demonstrated partial integration, such as encapsulating nucleotides and lipid vesicles, but no system has yet successfully combined replication, metabolism, and compartmentalization into a single, evolving entity. This lack of a fully integrated prebiotic system remains a central barrier to how life, could have emerged from chemistry alone. The most frequent way that evolutionists respond to these problems is complete dismissal. In other words, when presented with these problems, they deny that they are even problems. In other words, they will proceed as if none of these problems exist and that there is nothing to hinder abiogenesis from happening. Their primary response to the problem of statistics is to, without actually ever demonstrating that the calculations are wrong, simply denounce them as being based on false assumptions and/or erroneous calculations. They will never actually go through the calculations and demonstrate why any particular set of calculations is wrong. Their primary response to arguments about thermodynamics is to stick to the second law as the argument. They then throw out the fact that the Earth is an open system, and therefore entropy can decrease. They completely ignore the principle that actually explained what would happen in a prebiotic open system. They will sometimes go on to argue that life is inevitable because it is an efficient way of dissipating entropy by way of heat. They literally act as if adding energy to a system magically reduces its entropy. They tend to completely ignore the fact that bombs add a lot of energy to what they are blowing up, while greatly increasing its entropy. The primary response to arguments about chemistry is to deny that any of the problems actually exist. They've even gone as far as insisting that the junk byproducts actually help future reactions. Some of their responses also include insulting one of the primary sources of this information, Dr. James Tour. One of the things that the proposed models for abiogenesis have in common is that none of them even try to deal with many of these problems. They just seem to think that they will work somehow, and that the problems don't even exist. For example, it would be interesting to see evolutionists actually take one of their models and run a statistical calculation that demonstrates a realistic probability of life occurring at least once in the history of the universe. I can see 4 possible explanations for this. The first and least likely is that they have actually run such calculations and cannot make them work. This is not to say that they've made the calculations and have gotten them to work. After all, evolutionists would be beating creationists over the head with the results. Rather, it is a case of they can't get it to work and therefore have abandoned any effort to even try. The second is that the notion of abiogenesis is so ingrained in the naturalistic thinking of those doing the research that they are so convinced that it happened that they see it as a waste of time to even answer such objections. The third is that they don't even know that such objections exist, or if they do, they have such a gross misunderstanding of them that they don't believe that they are even worth answering in their models. The fourth and most likely reason is a combination of the second and third. That is, naturalistic thinking is so ingrained that any objections that they hear about are summarily dismissed and even distorted if necessary. They also try to claim that the lipid spheres that have been produced are actually living things. Lipid spheres, also called vesicles, are a key concept in abiogenesis research because they provide a simple model for how early life could have achieved compartmentalization, a crucial step toward forming the first cells. These spheres form spontaneously when amphiphilic molecules, like fatty acids, self-assemble in water, creating a hollow structure with a hydrophobic interior and hydrophilic exterior. This structure can encapsulate other molecules, such as nucleotides or small peptides, protecting them from dilution and degradation while allowing some exchange with the environment. Lipid spheres are significant for several reasons. Compartmentalization. They create microenvironments where chemical reactions can occur more efficiently. Concentration. They help localize monomers and polymers, addressing the dilution problem in prebiotic oceans. Dynamic properties. They can grow, divide, and fuse, providing a primitive mechanism for replication and inheritance. However, there are limitations. Prebiotic vesicles are fragile, sensitive to salts and temperature, and lack the selective transport mechanisms of modern cell membranes. This is the closest that they have managed to come to anything even remotely similar to a living cell. However, it still has many problems with regard to making the transition to an actual living cell. This is because it lacks the machinery and information contained in living cells. Consequently, you still have most of the same problems. Pretty much the only solutions it really solves are creating a compartmentalized environment where the concentrations of prebiotic chemicals can be adequate to at least do something. Would the creation of life in the lab prove abiogenesis? Not even close. Now it would be claimed as proving abiogenesis, but think of the reality. If scientists ever managed to produce a bacterial cell in their laboratories, what would it take to do so? It would take some very intelligent men, using some highly intelligently designed equipment resulting from many years of research to accomplish it. It would only end up supporting intelligent design in reality. The problems listed here with abiogenesis are just part of the picture. When you really look into it, the effort is not really driven by science, but by a 100% atheistic desire to remove God from the picture. There are all sorts of reasons to see why abiogenic research is a waste of time and money. In fact, there's only one reason for not concluding based on what we already know that abiogenesis is impossible. That reason is that it is an absolute demand of atheistic thinking. There are people in this world who desperately want to remove God from the picture. They desperately want to explain how we got here apart from God. This effort started more than 200 years ago, but reality keeps insisting God exists and He is the creator. However, these people will not only reject all evidence for God and His identity as the creator, but they will waste uncountable amounts of time and money in the effort to try regardless of the evidence.